thesis. He is fellow of the European Association for Artificial Intelligence Science 2012 and that of the Academia Europea Science 2018. He has been awarded the 2013 Spanish National Prize in Computer Science in the prize of the Spanish Association for Artificial Intelligence in 2018. In 2020, he has received the Amity Research Award in Machine Learning in New Delhi. His research interests are primarily in the areas of probabilistic graphic models, meta-heuristic for optimization, data science, classification models, and real application, such as biomedicine, bioinformatics, neuroscience, Industry 4.0, and sport. I leave you with Professor Pedro Larrañaga.
So thank you very much, Gustavo, for this kind presentation. It's for me a pleasure to be to participate as a keynote speaker in this MICAI 2020. And today talks is about Bayesian network in neuroscience, and I would like to show you part of the work that we have done during the last 10, 12 years in this topic of neuroscience, mainly in two big projects. One from the Spanish Ministry of Science and Innovation is the Cajal Blue Brain project, and the second one from the European Commission is the so-called Human Brain project. So the outline of the talk is as follows. I will introduce the topic, especially machine learning, several things, several issues related with machine learning. And then for those in the audience that are not familiarized with Bayesian networks, several main concepts on this paradigm, Bayesian networks. Then we will present two problems in neuroanatomy and two problems in neurodegenerative diseases. In all these problems, my main objective is to show you challenging tasks from the point of view of machine learning. And I will conclude with a general discussion about the use of Bayesian network in this topic, and showing some references that I will use during the talk. So you probably know this book of Domingos, the master algorithm, where Domingos speaks about the 5 types in machine learning. And for each type, he defined the main algorithms. For example, in evolutionary, he speaks about genetic programming. We can add genetic algorithms as the main algorithm. In connectionist, of course, backpropagation, that is the main algorithm in neural networks or even in deep neural networks. In symbolics, this is a field that we can think about something that is the old-fashioned artificial intelligence. And then we have analogizer with kernel machines and Bayesian, that is the main topic of this talk, with probabilistic inference. So I would like to show you this slide where we can see different tasks in machine learning, and with for each task we can see also the main algorithm. the most important algorithm. For example, in clustering, when we speak about non-probabilistic clustering, we have agglomerative hierarchical clustering or k-means from the point of view of partitional clustering. Or when we speak about probabilistic clustering, we can think about finite mixture models as one possibility. We can speak about multi-view clustering or even about supervised classification with non-probabilistic, probabilistic, or even meta-classifiers. Or we can speak when we try to predict several class variables at the same time. We can speak about multidimensional classifiers, or even about more sophisticated tasks in machine learning as anomaly detection, discovery association, feature subset selection, or even multi-output regression. As you can see, there are some ad hoc methods for each of these tasks, but there is something in common, that is that there are some approaches based on Bayesian networks able to provide some solution to each of these tasks. So before to introduce Bayesian network in a naive way, I would like to show you these artificial intelligence principles defined in the Asilomar conference by the Future of Life Institute 3 years ago. And here we can see the 7th and the 8th principles that are related with transparency. At the same time, we see in this recent paper by Rudin in Nature Machine Intelligence that the main message for her was to stop explaining black box machine learning models and use interpretable models instead. So today we will use, or we will speak about the application of one of these interpretable models, Bayesian networks, in a hot topic that is computational neuroscience. So what is a Bayesian network? I will try to avoid a very technical definition. I prefer to introduce this paradigm with one example. This is one hypothetical example that we introduced in this paper in Frontiers in Computational Neuroscience 6 years ago. It refers to something that we call the risk of dementia. Let's consider 5 variables, as you can see here in the graph: age, neural atrophy, stroke, dementia, and paralysis. And you can see that these nodes, or some of these nodes, are connected with other nodes. For example, age is connected with an arc to neural atrophy and stroke. In Bayesian network, we said, using the standard terminology, we said that age is the parent of neural atrophy and stroke, and these 2 variables are parents of dementia, and the second one, stroke, is parent of paralysis. But apart from this relationship between the variables, in this case, this relation is speak about causality. Apart from this graph, we also need some quantitative part in our Bayesian network model. So we need these conditional probability tables. As we can see here, for age, we only need a prior distribution. By the way, all variables I didn't mention before, but all variables are binary. In this case, age, of course, is a continuous variable, but let's assume that is discretized as a binary variable. As age is the root of the network, we only need a prior distribution for age. But for the case of dementia, the CPT, the conditional probability table, is a table with 8 entries. In each entry, we have the conditional probability of each value, each possible value of dementia given any possible combination of neural atrophy and stroke. Using this representation, we can factorize the joint probability distribution over the 5 variables in the network. As you can see here in the bottom part of the slides, we have the prior distribution, or the prior probability distribution of age times the conditional of neural atrophy given age times the conditional of stroke given age times the conditional of dementia given neural atrophy and stroke. And finally, the last factor is the conditional probability of paralysis given stroke. So once this network, this graph, this directed acyclic graph, and the conditional probabilities is given, the network can be used for making any kind of probabilistic reasoning, from diagnostics to causal inference, or even to predictive inference, or for abductive inference. I will try to show you in the next slides the main concept underlying Bayesian network. And this main concept is conditional independency among triplets of variables. So let's start with this situation where we don't introduce any evidence in the network. Here we are representing the marginal distribution for each of the 5 variables. And let's introduce our first evidence. Our first evidence is a stroke equal to yes. So we introduce this evidence, and as a result of this evidence, when we consider this evidence along, or transmit this evidence along the network, we can see that almost all the probability distribution of the variables, of the rest of the variables, have changed. For example, we had 75, 12, 17, and 11, this prior distribution. And now, as you can see, this probability distribution is or are completely different only after considering that the stroke is equal to yes. Our second evidence will be neural atrophy equal to yes. And in this case, I suppose that the probability distribution of age and dementia are now different. So they were 94 and 52. Now we have 99 and 96. But notice that the probability distribution of paralysis, 75, remains the same as before. And finally, and in order to present or understand this idea of conditional independency, let's consider that our patient was a young patient. So I will introduce this evidence in the network. So now we have a young with neural atrophy yes and stroke yes. By doing this, we can check, but the probability distribution of dementia and paralysis remain unchanged. They are the same as in the previous slides, 96 and 75. So what is the meaning of this? Of course, there is not a mathematical proof, or it's only checking that if we introduce this evidence of age equal to young, once neural atrophy and stroke has been fixed, this property of neural atrophy and stroke, this situation in the DAG, is able to block all the information from age to dementia and paralysis. Technically, we can say that age and dementia and paralysis are conditionally independent given neural atrophy and stroke. And this conditional independency between triplets of variables is the main semantic underlying this field of Bayesian networks. More technically, Bayesian network has two components: a graphical structure that is a directed acyclic graph, where the vertices represent the variables and the arcs represent conditional dependencies or conditional independencies. And we also need a set of parameters, as I mentioned before, in the CPT. By using this, we can factorize the joint probability distribution, as we represent here with as many product or as many factors as a variable in the network. For each variable, we have the conditional probability of the variable given his parents in the network. We can obtain this network. We can do with this network any kind of probabilistic inference, as I mentioned before. We can introduce some evidence here in the green nodes and try to see how this evidence, after propagating the evidence through the network, change the probability distribution of the rest of the variables. There are some exact and approximate algorithms that are able to do this kind of tasks. In addition to this, there are methods, learning methods, able to automatically transform a data set into a Bayesian network structure, and to compute also the parameters for this structure. And this can be done using two main approaches. One is detecting conditional independencies by the use of hypothesis test. And the second main approach is with the use of score plus search methods. Here we have one schema about the type of search spaces, about the different kinds of scores, and the way to find in an intelligent manner in these very huge spaces. To finish with this part, clarify that parameter learning used to be done using maximum likelihood estimation, or even using Bayesian estimation. In this case, we need to define a conjugate probability distribution with respect to the categorical or the Gaussian distribution. Okay, so let's start with the applications of this paradigm, Bayesian network, that from my point of view is quite transparent, quite interpretable. And let's see application of this into main topics in neuroscience. One is neuroanatomy, and the second one is neurodegenerative diseases. I have selected four applications that we have developed during the last years. In all the applications, my main idea, as this is a Congress on Artificial Intelligence, is to emphasize the challenging problems that we have in neuroscience for machine learning. So I will start introducing some concept in neuroscience in general, but before this, some words about the Human Brain Project. It's a very long project. We will finish in 3 years. And the main idea of this project is to work together in neuroscience, medicine, and computing. From the point of view of neuroscience, the idea is to approach this problem in one holistic way, with a multi-level view of the brain, starting from genome, transcriptome, proteome, finishing in the whole brain. The idea is that here there is more than 150 institutions, almost all of them are European institutions, working together with these three main objectives. Probably you are aware about some characteristic of the human brain. I will show you in a very fast way. Our brain is composed by 4 lobes: the frontal, the parietal, the occipital, and the temporal lobe. And you have here some numbers corresponding to a normal or a healthy brain. On the other hand, in the cerebral cortex, we can consider or distinguish 6 different layers, as we can see in the right part of the figure. And from one microscopic point of view, the brain is composed by neurons, blood vessels, and glial cells. And according to Santiago Ramón y Cajal, and according to his neuron doctrine, the neuron is the basic structural and functional unit of this nervous system. Nowadays, estimation quantify in 86,000 million of neurons, the number of neurons in a normal brain. So, sorry. Yeah. So in a neuron, we can distinguish 3 main parts. One is the dendritics, as we can see in this part. The other one is the cell body, also known as soma. And then we also have in each of our neuron, we have the axon who connect different neurons. Okay, so let's start with our first problem. Our first problem refers to the clustering of something called dendritic spines. These dendritic spines, as you can see in this A figure, they are small protuberances in these dendrites. And according to the literature, depending on the shape of these dendritic spines and the distribution of these dendritic spines along the dendrites, we can speak about a normal or an abnormal brain. So here our main interest is in clustering these spines from one objective point of view. The standard clustering approach in this field was proposed a long time ago by Peters and Kaiser-Abramov, and only consider 3 types of spines, as we can see here: thin, Murrow, or stubby. So we tried to develop, our objective was to develop a more objective clustering of spines. For doing this, we obtained some electronic digital representation of these spines using microscopic electronic microscopy. And after repairing some of these spines, we finally obtained a reconstruction of about 8,000 spines, corresponding to humans aged 40 and 85, All these spines are from this part of the cerebral cortex. All of them correspond to pyramidal neuron. Perhaps is the time to clarify that our neurons are mainly of two types. We have something that is called pyramidal neurons, and we have other type of neurons that are interneurons. Pyramidal neurons are excitatory neurons, and they don't have much interest in the sense that they are almost equal in shape. But on the other hand, interneurons are they present a completely different shape, as we will see in our second application. But let's continue with this, because with each of these 7,900 spines, we characterize each of them using 54 morphological features. And in some of these features, they were directional variables. I mean that with directional variables, sometimes called circular variables, we are speaking about mainly about angles in the dendritic tree. So in this case, we can use normal distribution or Gaussian distributions, because one angle that is equal to 1 degree and another angle that is equal to 359 degrees are very, very similar. So we need to use other kind of probability distributions that people have developed for these directional variables, as for example von Mises distribution or von Mises-Fisher distribution. And this is the way that we developed this approach in one recent paper in this publication. But to have a general idea of this, we approach this clustering problem using a multivariate mixture model with as many cluster as component in our mixture model. So each of the component of this mixture model was a Gaussian, or if we incorporate the directional variables, as in this recent paper, we have two types of variables, longitudinal or standard variables with a Gaussian distribution, and at the same time in the Bayesian network we also have directional variables. So as a result of this and developing a score plus search approach, as I mentioned before, we determine the structure of this model, of this mixture of Gaussian Bayesian network model by using the structural EM algorithm. So we use the expectation maximization algorithm, but in this case, at each step of the algorithm is able to change the structure, or to adapt the structure of the Gaussian Bayesian network. We finally obtain 6 clusters, and here we are representing a simulation of these virtual spines from each of the clusters. Okay, our second problem is in supervised classification, but as we will see now, is a special case of supervised classification, where the labels of the neurons, in this case interneurons, are not clear, or we can see that they are probabilistic labels in some sense. So let's speak about the problem. The problem refers to the different kind of interneurons. People speak about mainly 10 types of interneurons with these names: Arcade, Chandelier, Horsetails, Martinotti, common type. And each of these type of interneuron has his own representation. So this is a chandelier, or this is a horsetail, and so on. So our idea here is to check the categorization of several experts from different labs in different countries, mainly in China, United States, and Europe. And we consider 42 experts. We develop a web-based system. We saw around 300 interneurons to each of the experts, and the experts were asked about these 6 variables. The six is not important here, so I will speak about these 5 variables. The first one is the first and the second one refers about the position of the dendrites. We can classify this as intralaminar and translaminar, or as intracolumnar or transcolumnar. The next 2 variables refers about the position of the soma, if it is centered or displaced. Finally, the 5th variable, that is our main interest, refers to the type of interneuron. So it means that at the end of the day, we have something like this. We have a table with as many rows as neurons, with a lot of predictor variables that automatically we obtain from the morphological aspect of the neuron. Here we will see later on, but we are speaking about 3,000 morphological features. And then we have the class variable of each of the 42 experts, with C1, remember, refers to intralaminar or translaminar, is a binary class variable. C2 is also binary, and so on. Finally, our main interest is here in the type of interneuron. The main problem is that the experts, they don't agree about these 5 variables. This work was published in a journal in the Nature Review Neuroscience, and in this representation we can see the inter-expert agreement. Here we have C5, the different types of interneuron, and we can see some interneurons, as for example Martinotti, where the agreement is between high and medium, but we also have at the same time several interneuron types: common type, common basket, Cajal ratios, Arcade, and others, where the inter-expert agreement is low. In order to show you this with a Bayesian network, here I represent the Bayesian network corresponding to expert number 16 and expert number 27. This network have been obtained from data. So we introduce the evidence of Martinotti in both cases, And as we can see, when this evidence is introduced, the probability distribution of the rest of the variables change accordingly. But we see that, for example, the probability of translaminar is quite similar in both cases. The probability of transcolumnar is quite similar. Displays probability is quite similar. The situation is completely different with a common basket. For the same expert introducing a common basket, we can compare the probability distribution of intracolumnar completely different, or even this one of centered or intralaminar. Okay, so this shows that there are some interneurons where the degree of agreement is very, very low. So, but remember that our main objective was to classify this interneuron, and I will present here 3 different ways to do this, because our main problem is that our label is not clear. We have 42 experts, and each expert decide one label. These 3 approaches, I will concentrate in the first 2. I will only mention some aspect of the last one. So in the first one, the neuron label will be considered as the most frequent cell type. So here we reduce the number of neurons. If you remember, we have around 320. Now we only consider those neurons that were visualized in 3D. We have about 3,000 predictor variables, morphological features. And here we are representing for each of the neuron the most frequent class. Martinotti for the first neuron, large basket for the second, and so on. We use different classifiers, as we can see here, from Gaussian naive Bayes to random trees. Different feature subset selection methods that in this problem plays a very important role, from univariate filtering to multivariate filtering. And by doing this, our results are not very good. We are able to, in this multi-class problem with 10 possible values for C5, we are only able to obtain accuracies of 62%. So trying to improve this result, now we will consider different label reliability thresholds. And in this case, the idea is that we will consider, first of all, a small number of features as a result of the multivariate filtering in the previous paper. And we have these predictor variables. We also have these 4 class variables that can play the role of predictor variables, and our class variable is this C5. But between brackets, you can see the number of experts that agree in this value. So for example, in the first neuron, the most frequent class was CT with only 21 experts. So it means that if we introduce a threshold of 25 for this C5, this neuron, this first neuron, is not selected anymore, as only 21 experts agree on CT. So we can at the same time consider a threshold for the C1, C4 variables, here represented by 21, or in these other possibilities where the morphological features and at the same time C1, C4 are predictor variables, we can establish two types of thresholds in C5 and in C1, C4. So as you can imagine, when we increase the number of these thresholds, the number of neurons is reduced dramatically. And after using some Bayesian network classifier, different Bayesian network classifier, starting from naive Bayes or using selective naive Bayes, or even attribute weighted naive Bayes, where the algorithm is able to compute automatically the exponent number here, or tree-augmented naive Bayes, we obtain some results that can be seen in this slide. As you can see, here we have the accuracy that have been estimated using a 5-fold cross-validation scheme, and the best results are obtained when all the morphological features and also the class variables are used as predictor variables. In order to show you the transparency and the interpretability that I mentioned before for Bayesian network, in this case Bayesian classifier, let's consider this toy example where we have binary class, large basket and Martinotti. We try to predict the class using these 5 predictor variables in a tree-augmented naive Bayes scheme. If I introduce some evidence on total length, and I introduce an evidence that the total length here is medium total length. In this case, we can see that the Martinotti posterior probability increases until 62%. And now until when we introduce apart from this total length, when we introduce the evidence about the maximum Euclidean distance in the neuron, this posterior probability increases, and finally, when we have 1, 2, and 3 evidence, this is enough in order to classify almost sure this interneuron as Martinotti. Okay, this is the other possibility that I will only mention. It was developed by one of our PhD students and correspond to generalization of the expectation maximization. Is called the generalized expectation maximization, where here we are considering a probabilistic label in each of the 10 interneuron types. So instead of using a class label, here what we say is that this neuron, according to 4 of the experts, is type 1, let's say ARCAD. According to 7 of the experts, is type 2, let's say Martinotti, and so on. Okay, I would like to finish this neuroanatomy applications with a short comment on this recent paper in Nature Neuroscience, where several people in our research group included, we agree on the use of transcriptome for classifying cell types. So in the previous application, we have seen the use of morphological feature for this task. There is a lot of people also working in electrophysiology, but nowadays it is possible to consider a more interesting approach based on transcriptome. To tell you the truth, people from the Allen Institute is pushing about this direction. Okay, I will change my application domain, and I will present again two challenging problems in neurodegenerative diseases, challenging from the point of view of machine learning. The first one refers to Alzheimer's disease, the second one to Parkinson's disease. And as you probably know, the effect of this disease are related with memory disorder, cognitive decline, and loss of autonomy. And in this application, we will use some microarray data that was presented for the first time by Small et al. 15 years ago. So they refer, this microarray data refers to the most vulnerable to Alzheimer's disease brain site that correspond to the entorhinal cortex. This entorhinal cortex is located close to the hippocampus, and Small et al. Work with 12 tissues where they obtain the expression level of more than 7,000 genes. The main problem here is the small sample size, only 6 Alzheimer's disease brains and only 6 control brains. And our objective was to reanalyze this data, trying to obtain robustness from this small sample size. We aim to obtain new explicit biological relationships and genes related with the Alzheimer's disease that has not been previously reported in the literature. And to do this, we use one type of classifier called K-dependency Bayesian classifier. This generalized the tree augmented naive Bayes, allowing to each predictor variable until K parents apart from the class variable. But the main idea in order to obtain robustness in the model is to work in a resampling way using bootstrap sampling. And we try to obtain something like a consensus network in the final gene interaction network. And for obtaining this, our idea is to establish a reliability threshold T and only select those arcs occurring more than T times. As we can see in this example, if T is equal to 800, we will only retain this arc that appear 800 times. If we decrease the value of T, the network is more and more dense. And in our specific application with 12 samples and with 4 dB, K is equal to 4, with 10,000 bootstrap samples and a T a threshold of 1,000, we obtain these two directed acyclic graphs that are not connected. And after checking the different genes in this gene interaction network, we see that, for example, this gene was reported in the literature related with muscular atrophies. This another gene with mental retardation, Huntington disease, bipolar disaster, and in the second network we have this gene related to Parkinson disease. So to finish with the 4 application in neurodegenerative diseases, let's see how to transform 2 popular quality of life instruments to measure the degree of disability in Parkinson disease patient. These 2 quality of life instruments are this one, the 39 items Parkinson disease questionnaire, that is an specific instruments, as we can see in the slide, capture the patient perception of her illness, covering 8 different dimensions, as we can see here, from mobility to communication or bodily discomfort. Each of the dimension contains several questions, in total 39 questions, and for each question we have 5 possibilities from never to always. The other quality of life instrument is the European quality of life instruments. This is not specific to Parkinson disease, it's a generic instrument, and different nations, especially in Europe, use it for clinical and economical appraisal. And here we have 5 concepts, from mobility to anxiety or depression. For each of the question, there are 3 possibilities to answer. So in an abstract way, we have this problem. We have information about some patients, around 400 patients. For all these patients, we have this information about the Parkinson disease questionnaire, 49 variables that will play the role of predictor variables, and at the same time we have 5 class variables, the variables in the European Quality of Life questionnaire. And this is like this because this part of the information can be filled by the patient or by some relative of the patient. In machine learning, from a machine learning point of view, we are speaking about a multidimensional classification problem, where we try to predict these 5 class variables. And if we try to do this from one probabilistic point of view, we are speaking in something that was known as a multiple diagnosis problem. One direct approach will be to search for the most probable explanation, that is for the combination of class values, in this case European quality of life, that maximize the posterior probability of this class variable given these 39 variables. Using Bayes' rule, we can write like this, but in the most simple case where all variables are assumed to be binary, remember that in our application they are not binary, they have 5 or 3 possible values. But in any case, in the most simple case, the number of parameters using this approach, this naive approach, is exponential in the number of predictor variables M and the number of class variable to be predicted. So we decide to apply this multidimensional Bayesian network classifier introduced 14 years ago. And in this type of classifier, we consider the network or the graph composed by 3 subgraphs: the class subgraph, representing the relationship between the class variables; the feature subgraph, representing this relationship; and something that we call the bridge subgraph, representing the arcs between European quality of life variables and PDQ 39. More specifically, as we are interested in computing the most probable explanation, this is a problem that is NP-hard in Bayesian network, and we try to alleviate this computational border for computing this most probable explanation, and we define one type of multidimensional Bayesian classifier called class bridge decomposable MBCs. With this class bridge decomposable that verify these 2 properties, we are able to alleviate the computational border for computing the most probable explanation. For example, we can compute the MPE for this part of the graph. We can compute the MPE for the second part of the graph, and the final solution will be the concatenation of the these two previous subparts. So we use this approach in collaboration with Abbott Laboratories in Switzerland, with about 500 Parkinson patients, and we compare the behavior of these MBCs, multidimensional Bayesian classifier, to 2 different types: the class bridge that I mentioned in the previous slides, and another proposal based on the Markov blanket. We compare this with state-of-the-art in this kind of problems, using mean accuracy and the exact accuracy. And as we can see, the result after a 5-fold cross-validation scheme were very, very good for the multidimensional Bayesian classifier approach. And in addition to this good result, we were also able to obtain the model, is one explicit representation of the model. As you can see here, the algorithm was able to remove some of the 39 predictor variables, selecting only this small number of predictor variables. And we are using 3 I didn't mention before, sorry, but this class bridge decomposable is based on the concept of maximal connected component, and here we have in this problem 3 maximal connected component. So we obtain this decomposition, and what is more important, we obtain some relationship among the variables that are able to produce new knowledge for the neuroscientist. So all these 4 examples that we have seen with Bayesian networks, and other examples with Bayesian networks or other kind of machine learning and statistical models can be seen in this book that it is expected to be published by Cambridge University Press in November 2020. So I conclude saying that Bayesian network is a tool for data science. is a machine learning paradigm based on probability theory and graph theory with several advantages, between them transparency and interpretability. This is very interesting for nowadays explainable artificial intelligence. The paradigm has a exact inference algorithm providing any or almost any kind of reasoning inside the network, reasoning for predictive diagnosis, intercausal or abductive inference. There is also a plethora of learning algorithms from data for different tasks, such as clustering, multi-view, supervised classification, multidimensional classification, or more complex tasks as anomaly detection, discovery, association, feature subset selection, or multi-output regression. Both inference and learning algorithms can be adapted for temporal and data stream scenarios that are very common in nowadays application. And of course, as the paradigm is transparent and interpretable, it is possible to obtain new knowledge in a given domain. Here we have seen in neuroanatomy and neurodegenerative diseases. So to finish, I can share with you this long list of references that we have used along the talk. And that's all. Thank you very much for your attention.
Thank you. Thank you very much, Pedro, for your interesting talk. We start with the question session. Let me check the questions. We had one question. Can you see the question, Pedro?
Okay. Okay. I suppose that is
you can explain the reason to consider
yes.
only.
Yeah, I suppose that the question is related with this part, I suppose.
Okay.
Is the only the question was about 5 predictor variables, Gustavo?
Yes.
Yeah, in this case, this is probably the question refers to this. This is a toy example, as I mentioned. This is a toy example, and the main objective of these 3 slides is to show you that Bayesian networks are interpretable and transparent, but there is not any other reason. And the second part of the question was about who was the most important, if I well understand, the most important predictor variable. To tell you the truth, with these slides, I can answer the question because it wasn't my objective. But in any case, you can develop some method to answer to your question. If you try to work in one sequential way, adding one variable at a time and selecting at each step in a greedy way what is the most informative variable, you can do it. Yes, of course.
Okay, thank you. There are another question. Can you see the question?
Yes. Yeah, it's a very interesting question. So it's something that is this course of dimensionality, or this situation with very few instances and a lot of variables. That in one of my examples, I spoke about 3,000 morphological features, and only around 200, 300 instances is very common in neuroscience in this case, or even in computational biology. Also in the problem of Alzheimer's disease, it was only 12 instances and more than 700 or 7,000 features. So this is a common situation, and the question is how to select what is perhaps is the most appropriate method, or how to approach feature subset selection in this situation. So I would like to say that in this situation I will recommend the use of filtering, perhaps an hybrid approach composed by a filtering at the first step, univariate filtering or multivariate filtering. There is a lot of possibilities: univariate filtering with mutual information, gain ratio, and so on; multivariate filtering with relief or even correlation feature selection. And after this, the use of this filtering, that is, these methods are not very computationally demanding, the output of this method will be the input for a wrapper approach. So what people do in this situation is to propose one hybrid between filter and wrapper. First filter, and the output of the filter is the input of a wrapper approach.
Okay. Thank you, Pedro. On behalf of Mexican Society of Artificial Intelligence, I want to you thanks for the keynote talk, and give you this diploma.
Okay, thank you very much.
Certificate.
It has been my pleasure to collaborate with you, and I hope that in the near future we can see each other, not only with these technological things, but physical. Okay, so thank you very much.
Thank you very much, Pedro. Thank you very much for the attendance. We continue with the program of MICA conference. Thank you very much.
Thank you.